Important regions inside the proteins are necessary for drug discovery phases. Now, when we talk about these important regions are important points inside the proteins, we talk about two things. Number one is with respect to structures, and the other one is with respect to the functions of that particular protein. Now, if we describe our ultimate goal for drug discovery, what is our ultimate goal of drug discovery, and particularly about target identification? The ultimate goal is to identify those relevant targets which can be used for causing some disease and help us to find out progression of that disease, and finally to make some drugs or ligands for that particular disease. Now, based on this definition, what we have got so far in human genome project, or based on human genomes only, we have roughly 3000 genes which can be used for this particular purpose, which is basically discovering or making some target for drug discovery. On the other hand, total number of genes discovered for human genes are 30,000. So what we can see is that as compared to 30,000 genes in total, we have only 3,000 genes which can be used for making some drug. If it is alone based on genes only, then might be it would be acceptable. But the unfortunate thing is that we have other macromolecules also, such as we have many thousands or hundreds of proteins available. So genes plus proteins, their number is very enormous or large. But when we talk about those genes or proteins which are used for drugs or which can be used as drug targets, it's very few. So we want to fill this gap between provided genes or proteins and druggable genes and proteins. So for that purpose, to identify or to fill this gap, we have several things available. We will start from homology modeling. So what are homologous, or what is homology modeling? In which we have some database of already provided proteins or genes, and then we want to apply or assign, or using multiple sequence alignment or different algorithms, we want to align newly discovered proteins or genes with the already discovered proteins or genes. So for whenever we talk about proteins, we have a concept called fold recognition, which basically helps us to identify different families, superfamilies, and subfamilies inside the protein sequences. So if we have this information and if we assign or align it with the already stored information, we can have very good data structures for finding or for annotating newly discovered proteins. One example for this thing is that when we have HIV inhibitors, which are basically used for the cure or drug or medications against the HIV/AIDS type diseases. So what happens is, if we talk about this homology modeling alone, it would be tough task. But the good thing is we have several tools available which help us to find or complete this task on our behalf. Number one tool, which is I have provided three tools here. So first tool is basically a modeler. So what is the purpose of modeler? Basically, it usually aligns different sequences using multiple sequence alignment. An example can be it can easily align 100 amino acid sequences at one go. So what would be the advantage? If they can be aligned successfully, then we can say that they are homologous to each other. They are from same ancestor, or there are much similarities against these sequences. The other one is threading. Threading is basically a template based approach. So what is our input, and what we want to do is that we have a basically a sequence which is already discovered, and then an undiscovered sequence or a new sequence. So what we will do, we will put the newly discovered sequence as a probe into the already discovered sequence, and if there is some matching, then we will say that they are homologous, or there is a homologous modeling method available for these two sequences. Finally, we have ab initio methods. In this ab initio method, basically we don't have a prior information about the newly discovered structures. So if there is no prior information, we don't know what is this structure is, then we have different statistical methods which we can use for getting the annotation of these structures. And logically what we do is we start with very short segments, because shortest to capture the shortest thing is easier. So start from the shortest, then try to merge them or combine shorter segments into bigger or larger segments and make the complete results. Then we have how can, as we have already said, for a protein structure, we have two things. One is structure information and the other one is functional information. Now, how can we predict functional regions or functional attributes of a protein structure? So there are four different methods available for this thing. Number one method is based on sequences itself. That based on the sequence structure which is provided in our macromolecule, we try to guess the functional information about that structure information. The other one is basically apolar conserved residues clustering. So basically what happens in clustering, that we don't know exact information about the residual parts, but what we do, we try to make clusters. Clusters means those boxes which have the same functioning. So macromolecules having same functioning are put in the same clusters, so we have different clusters at the end. Third one is basically conserved functional group analysis. So group analysis means that we put similar functions in groups and then we try to analyze them. What is basically, if there is some difference, then we remove that particular macromolecule from that particular group. And finally we have sequence profile and neighboring residues. So in this one what happens, that we have a complete profile of the given sequence, and that profile is also dependent on its neighboring macromolecules. So if there is some change in neighboring macromolecule, it will affect the inner molecule also, and we have to capture these changes which are based on neighboring molecules. So finally, how we can discover structurally related macromolecules or structurally related proteins. For that purpose, we can have four options. So basically what we want to do in this particular part is, we have some unknown sequence and we have some known sequences, and we want to compare them, how we can align them or compare them, if they are similar, different, or if this is a new protein. So for that purpose we have four different methods. Number one is lookup method, in which we just compare the unknown sequence with the known sequence, and if there is similarity, then we say okay, they are homologous. Then we have fuzzy functional form. So fuzzy functional form, basically that is again similar to this comparison, but this is approximation only. So we are not 100% or dead sure that this sequence are similar, but approximately they are similar. Then we have this ProCat database. ProCat database, basically it is the database of catalysis or enzymes which are parts of proteins which help proteins in their functioning, but major information is related to these enzymes in this ProCat database. And then finally we have automated prediction. This automated prediction, basically in addition to structure, it helps us to identify functional information of proteins also. So these four methods are used for structurally related proteins, find how they are related to each other, and how to find unknown sequences from known sequences.